Okay.
Hey everyone, welcome. Welcome Diwakar. Thank you for agreeing to give a Build a Cell seminar, and you can get started.
Okay. My name is Diwakar Shukla. I'm an associate professor in chemical and biomolecular engineering, but I hold appointments across the campus in different departments, including chemistry, plant biology, bioengineering. I forget to list all of them. But it's also my research is very interdisciplinary, so we do a lot of work on different types of problems. So today, when I got invited for this seminar, I was thinking, what should I talk about? Just like a week before, I had put in a calendar, blocked the time to think about this seminar. And I thought we should basically talk about how interactions between different entities could be learned using AI-based methods. So my lab does a lot of computational simulations at a very large scale. But I'm not talking about that aspects. We also do experiments, very high throughput experiments as well now over the last few years. And so I'm not talking about that part. I'm just talking about one part of the work that I do, which is on AI. So uh, when I was thinking about this problem, I'll try to give you a few examples from my work about how can we learn like protein ligand, protein peptide interactions. And we all know that the protein ligand interactions are very, very important. They play a role in all sorts of cellular regulation of cellular behavior, physiology. And these interactions are really complex. So if we can learn them if we can learn them and learn them at a scale, just give the protein, give the ligand, and you can predict, let's say substrate specificity, or if you can give a sequence of a protein and a peptide, and it can quickly predict, interesting interaction points for these peptides and proteins. That would be really, really useful. There's a lot of work going on in this domain, but I'll show you some of our mostly unpublished work, which is currently under review. So at least it's, state of the art as of now. And maybe, in 6 months, someone will come with something cooler in AI, and then this will become old. So, you know, uh, and just to keep on going about why these interactions are so important, like, you have binding pockets that have evolved to stabilize all sorts of very specific types of ligands. Um, you also have a role of water in those, binding behavior. And then you can have a lot of different types of groups that can bind proteins as long as the key contacts, critical interactions are sort of, conserved. Now, if we look at the data, what kind of data is available? Uh, you know, this paper that I have cited here is from 2020. It has a nice distribution of how common are different types of ligands, for example, what kind of proteins are most common, what protein ligand complexes are available. If you look at it, there's approximately 20,000 unique protein ligand structures. Uh, I should not have used the word unique here. Probably around 20. There's a new data set called PDB Bind Plus now, which is like approximately like 22,000 now. And you can look at those examples from the PDB, right? But if you look at the ligands that are available in those, there are not that many ligands. Like most of the ligands are, your common um, common molecules that you find in cells. It will be ATP, it will be ADP, it will be NAD, all the heme, all sorts of very common molecules. They are more abundant. So it's difficult to, learn these interactions well. Although, people still make models, and then, they do comparisons and it looks okay. But as soon as you give something novel to these models, they usually fail. So, and it's very challenging even with cryo-EM and other, latest advancement in structural biology and with AlphaFold even, generating these data sets becomes a little bit tricky. So, what would we do? If you look at a traditional sort of a machine learning pipeline, there are opportunities to predict protein, ligand, protein, all sorts of interactions. Uh, you can think of modifying one of these steps. You do data preparation, model development, model training, evaluation. You can modify some of these steps, especially the data and the model development part, to actually leverage new kinds of information to augment your existing data sets. And this is very critical, in for other problems as well, as I've written here, that in chemistry we rarely have access to large data sets. So most of the times, what is really needed is a zero-shot prediction. So you just you have no data and you just want to leverage all the existing data and make an intelligent prediction about your problem. So I'll just give you a few examples from my work where we have tried to look at different strategies on how can we tackle this sort of low data problem for protein ligand specificity, protein peptide specificity. So the first example, this work is currently under reviews, going through this long cycle of review. This was a collaborative work with Professor Huimin Zhao, who is also a professor in chemical and biomolecular engineering at University of Illinois. And this project sort of started with the AI Institute at UFI, where we were. It's called Molecular Maker Lab Institute. So the idea is, how can we make molecules using AI and automated synthesis? So you need programs that can predict interactions, programs that can predict enzyme substrate pairs. So that was our motivation. If we want to use enzymes for synthesis of molecules, how can we readily predict unseen enzyme substrate interactions? So what I've shown you is like a big figure, very complicated figure from this manuscript, but it gives you a big summary of the key ideas. So you know you could be looking at a protein sequence, a substrate structure, and here. And then what we were able to do is we are able to do a very massive docking campaign where we are able to find millions of protein ligand pairs that have never been resolved in crystal structures. I think we ended up doing close to half a billion um, protein ligand docking um, uh, uh, tasks. And so we were able to get we were able, even if we filter out a large number of them, which were poor predictions, we take only high scoring ones. We can actually learn the interaction between the between the proteins and the ligands in this case. And so here we have built in some new message passing neural networks to learn the interaction between the enzymes and the proteins. And then there is at the end there is this cross attention where we are trying to the learn the basically the interactions between the enzymes and the ligands. So the cross attention is trying to focus the attention of the model on the residue pairs and the chemical groups that are interacting with each other. So it basically identifies the key interactions in the pocket, it zeroes in on them automatically, and then it gives you a very good quality prediction of the enzyme substrate pair. Now, so this is the overall idea, that you are leveraging uh, computational data to augment the missing experimental data. And the second idea was that if we can somehow learn the interaction between the enzymes and the substrate, then those interactions are much more fruitful in sort of zero-shot predictions of these uh, enzyme substrate pairs. So now, how do we do this at a scale? So we wanted to first generate all the data that we could get on protein ligand pairs. So there are standard data sets like UniProt and Brenda that you can use. But we also went to the entire literature that we could find and did basically data mining. So there was an automatic identification of reactions from all the literature that we could get our hands on. So these are all the patents, all the research articles, the reports, anything that is available. We try to mine for reactions in those. And then the machine is automatically recognizing the reactions, enzymes, products, substrates, translating that into smiles, and then sort of preparing the data set for the model. So we call it the enzyme specificity data set as of now. Uh, and then you use this data to build things, and you also add in computational data to this data sets. So the hundreds of millions of dockings that were done, we can actually we also augmented that data into our big general data set for enzyme substrate pairs. Okay, so just to break this out quickly, um, uh, so we are using a lot of like web scraping tools like OSRA, which is optical recognition softwares, and they can generate all sorts of inputs from these publications. But then this data set is very sort of rough. Sometimes you don't get right predictions. So then what you need to do is actually clean this data set. So, uh, we also added additional data, as I said, from Brenda and Unipart. And after that, again, another cycle of cleaning up these data sets and data preparation, remove all the faulty data points, remove duplicates. It probably took like I would say a year to do this well. So now this data set is all available. We'll release it once the manuscript goes out. Uh, so as I said, one of the key idea was that we can actually look at the active structures of different proteins and different enzymes, and then actually start doing a massive docking thing. So you can also look, you can score your dock poses based on the affinity that you're getting, based on the distance from the critical residues, if you're already aware. And we use this Autodoc GPU version. And as I said, roughly, I think that numbers were like 500 million around that, which were which we were dockings that we did, but then we cleaned up, and I think it left with like 40% of that data was actually good quality and good affinity predictions. So now that you have this massive chemical libraries docked to all proteins with known structures, now the interactions could be done, right? Um, so you can also look at what were, but if you look at the different combinations, there were what are the different types of reactions, different types of substitutions, which different types of products, substrates that were available, and you can think of combinatorial combinations of these, and that's the sort of entire data set. Okay, so the model, the two key ideas were that, as I said earlier, is that cross-attention and the integration of the structure information were the two key ideas here. The cross-attention is essentially looking, as I again wants to say, is that you are looking at the sequence data and the structure data, and you're trying to do a cross-attention to identify the key residues that are important for this substrate specificity. And you're also doing an attention between the ligand and the protein, and then you are learning which chemical groups are interacting with which amino acids or local environment, which type of different types of local environments in the binding pocket. And then once you have done all this cross attention, then you can put in a very simple, a simple decision tree or a very shallow neural network, and then actually it can actually predict things pretty quickly. Okay. Uh, so what kind of inputs were used, just to get you an ideas? We started working with ESM2. It's a transformer-based model that is trained on just different protein sequences. And so what it is producing is only the sequence level information for the enzymes, right? So this is just an amino acid representation. But then you also have this structural information now. So there is what we have developed is in this interaction MPNN. The idea is that there's a message passing that happens. So you're representing your ligands and the amino acids in the pockets as a graph, and then you're looking at the interactions between the ligands and the protein. And from there, you can actually have an information about what kind of atoms, what kind of microenvironments actually lead to a recognition of certain types of subgroups in the molecules. Okay, so I think this was a good idea. And then, as I said, cross attention. This is a double cross attention. So the idea is that you are combining, you're doing two types of cross attention, where between the input embeddings, the structure to sequence, and sequence to ligand to structure. So different types of cross attentions are put together. Okay, so that's basically the entire sort of model. If I look at the model training, um, and how the different types of data sets are split. So the current, the current state of the art. So this is AUROC score for the specificity prediction. So the gray line here is the ESP, which is the current sort of state of the art model uh, that predicts enzyme substrate specificity. And the gray and the green are like two of our models that are developed in this work, uh, which gives you a very high accuracy. Like if you do a random split of your data, you're almost getting a 0.9 as your accuracy. It definitely drops if you are doing things like you remove the reaction. For example, you say, okay, in your training set, don't include the reaction that is carried out by this enzyme. So uh, you have no enzymes or substrate that are involved in that type of a reaction. So if there are like halogenases, so you remove all the halogenases from this data sets and all the substrate that are halogenated. So you have no information about that reaction. And then you train a model and you see how good the prediction is. And you can see its prediction is still reasonable. If you remove the enzyme completely, if you remove both reactions and the enzymes completely, then the accuracy keeps on going down. But in most cases, if you're working with known enzymes, you get a very high accuracy prediction, even with a sort of a zero shot. Uh, if you look at our full models, we just compared the last model here. We did ablation studies, and you can see that the structural information was very, very important. So if you remove this, you lose a lot of accuracy. We also tested this on 6 classes of enzymes, which had no data in our data set, very limited data in our data set. So these are nitrilases and phosphatase, thiolase, nitrulase. And there are some proteins which are like domains of unknown function. They are called DUF. So we were basically looking at these classes of enzymes because they have very limited amount of data. So we can do an experimental data collection. So we tried to collect some of the data for these different families with very limited data. And then you fine tune your current model uh, with this. And you can see that um, even with limited data, you're getting a reasonable accuracy for your model with random splits, very, very reasonable accuracy. And we can also look at another class where we also try to do the exact same thing with different types of halogenases. And here, what we were trying to see if we can. These are 4 different classes of types of halogenases for which we are generating experimental data. And these are thousands of reactions that were done for these enzymes. I just want to say that. So very, very big time-consuming, experimental work happened. And so we looked at these different types of different types of halogenases that are present, and then and then you can look at it if we compare it to ESP. Our model again this is the benchmark right now in terms of accuracy, and you can see that we can actually train very specific models for different types of halogenases and get a very high accuracy for these models. Okay, so. Um, And there was another result that I just want to show is that if you say, okay, I have an unknown substrate, how many? What was it? So this is basically trying to predict what is the accuracy what is the accuracy of if you have an enzyme and you know a substrate for it, whether the what is the accuracy of predicting that this substrate is actually, what is your accuracy for predicting whether this molecule is a substrate for this enzyme? Um, so if we basically find that if we can predict like 8 substrates, we actually find almost all the substrates in the top 8 substrates. And on roughly 70% of the time, you find that a substrate that is known substrate for an enzyme would basically be the top substrate, or would one of the top candidates. So if you want to do an experiments for finding out, a substrate for an enzyme, and if we give you 10 predictions, there's very high likelihood that you'll be able to find a substrate for that particular enzyme than just doing 10 experiments. So this was, I think, was also used here. So this is one model of where we have, you had limited amount of interaction data. You augmented a lot of computational data with the experimental data. Then you also had a nice sort of a model here, which is trying to leverage the interaction between the enzymes and the substrates to learn these unique behaviors. And then you can also have a very strong sort of validation on protein families with very limited amount of enzyme families with very limited amount of data. So this work hopefully should be out soon. This is going through a lot of cycles of review. We've just submitted a revision. And so hopefully this time we are done. I'll give you a couple more examples on these low data regime and some ideas on how these could be handled for enzyme substrate specificity. The first example is a peptide protein interaction type of a model where we are working with a very unique class of peptides called lasso peptides, and I work closely with one of my collaborators, Doug Mitchell, who is doing a lot of work uh, on lasso peptides and RIPs. So, what's that question? Maybe I can take. There are a few questions. Maybe I can. Do you want me to take it now, or do you want me to take it later?
Up to you. Most of the time, people answer all the questions at the end, but
okay, that's fine. Then I just follow the tradition. Okay, so I'll give you some examples, and then also, predicting some substrates for kinases and GPCRs, and using when you have absolutely zero data, how do you do it, right? So, give you one example from our work. So, peptides, I don't have to go into great detail. These are very important. That's the only thing I will say. Peptide drugs are becoming very, very common. Natural products, which are peptide natural products, are also being used, becoming very popular as drugs. Just like few weeks ago, there was a very nice paper that used lasopeptides as an antibiotic, and that could deal with the bacteria that are not amenable to a lot of current antibiotics. So the lasopeptides are becoming really popular, and they are very unique sort of a peptide where uh, you have a ring that is formed by a cyclase, which is an enzyme that comes in, binds the peptide, and closes this ring. And then you also have this loop that actually goes through the ring. So you are forming sort of a globular protein, and which is very, very tiny, less 20 amino acids, and it has a core and everything in it, and it can bind in like small pockets in enzymes and receptors, right? So there are some structures of the lasopeptides that are available, and this is one example of a lasopeptide. There's another work that recently came out, which had a lasso bound to another GPCR, I think. So from Osamu Nureki's group in Japan. So lasopeptides are becoming really popular. So what we were looking at, we were working initially. The idea was that we were looking at these lasso cyclases that show very high substrate tolerance, and uh, and then there are other lasso cyclases that have a very precise substrate specificity. They are going to work with only a very narrow set of peptides. So if you want to make lassos, the first thing that you want is you want which cyclase will actually tie the knot or the lasso, make the lasso fold for which sequence. So if you can have a list of lasso cyclases, then you can actually make, you given any sequence, you can convert it into a lasso if you know the right lasso cyclase for it, right? So that was our initial motivation, that if you want to make a lot of lasso peptides, how can we make it? What is the sort of toolkit of list of cyclases that could be leveraged for doing this, right? So there's a tool developed by, as I said, from my collaborator, Doug Mitchell's group, who used to be a professor here at UFI, but now has moved to Vanderbilt University. And he has developed this program called Rodeo. And the goal of the Rodeo was essentially that if it can actually do genome mining and find out biosynthetic gene clusters and actually identify different lasocyclases and lasopeptides that are cyclized by these things. Uh, so so what we initially thought of doing is, can we develop an ML model to predict the peptide substrate selectivity for fusilacin cyclase? Because fusilacin was a lasso that he was really like his lab discovered it, and he was really uh, it's very easy for him to work with it. So what we started doing is we wanted to actually start making a language model, because you can imagine that there is so much sequence data, there is ESM language and other types of protein language models that are present, right? So we can actually take just the peptide sequence data. And once we have the peptide sequence data, we can actually learn from the peptide sequence without any labels uh, the unique topology of the lasso. I think that it has a ring and it has a loop that goes through the ring. Um, and luckily, if you do all these bacteria genome mining of these bacteria, we were able to roughly get around 6,000 lasso cyclases and lasso peptides pairs. So there was, I would say, just enough data to actually work with AI and deep learning. Okay, so the idea here is you take an existing protein language model, you have your some data sets, and you can fine tune the existing language model to actually now represent this new class of proteins or peptides. So what we were basically doing, you have a protein language model, which is pre-trained by doing masking of the sequences. And then you can use this model to predict a lot of like downstream tasks. Okay. And ESM2 is the most popular choice because it's probably one of the best models. There's now ESM3 as well available. Okay. So what did we do? We tried a few other things here just to deal with the limited amount of data. We started doing active learning, and we also tried using the graduate students who have spent like 5 years studying lassos and lasso cyclases to actually help us in the active learning process. So the idea here was that we wanted the human in the loop type of a training where we used to have a graduate student and their advisor. They're picking out the substrates that would be most useful for learning. So we used to predict these substrate, and then they used to say whether this will be a good whether this is a substrate or not a substrate. And then the goal was that we have to be better than the someone who has spent 5 years working on fusilacin cyclase. So we were able to actually do only few cycles of this active learning, and we could easily beat the experts on this topic. So the idea here is that you trained a peptide specific language model. Well, what you do is you hide certain part of the sequence, you give the remaining part of the sequence, and then you learn the missing part, right? That's the entire idea before for mass language modeling, right? And then once you get from there, you can get a representation of the peptide. You can train your downstream models. You can predict whether a particular peptide can form a lasso or not a lasso. Then you select some samples for testing, and then you can add that data back to your data set and retrain the model. Okay. So we have a lot of label data for different sequences, and then we get embeddings and we predict this. Okay. So we developed like two different types of models here. One was a peptide ESM, and the other is the lasso ESM. The lasso ASM paper should come out very, very soon. I can also say that in Nature Communications, it's just very minor edits are needed, but so this work will be available. But it's also available as a bioarchive, so anybody can read this paper. So we developed two different types of language models, which were like the peptides language model and a lasso peptide language model. And the idea there was that you have a lot of data from UniProt for lots of proteins. You can also take um, one of the tricks that's become very common now that we have also utilized for peptide protein interaction is that you take protein-protein complexes and then you can cut fragments of the protein interaction surface where you're cutting one protein and assuming that it's a peptide. So you take a continuous fragment that is interacting with the surface, and then you can call it a peptide, and then you give that data to the model to learn the protein peptide interactions. So that way you can augment your there's very limited amount of the protein peptide data, but there is, I would say, enough of protein interaction data. And then if you generate this, what you say, this augmented data for protein peptide interaction learning, then you can actually learn these interactions really, really well. So then you get very, very large data sets from a very limited number of protein interaction complexes. Okay. So without going into details of this, I'll just say we trained a lot of different models, and the lasso ESM sort of outperforms almost all of them. And these are some numbers just to say them. They look all very reasonable. These are the definitions of the different sort of benchmark. What you can see from the models is that if I look at the substrate and the non-substrate, we do a dimensionality reduction, although TSNE is, we can use this as a representation of whether the substrate and non-substrates, something's a peptide that is a lasso and a peptide that is not a lasso, can we sort of put them into different dimensions or different parts of this landscapes. And you can see that vanilla ASM doesn't have much information about peptides. Peptide ESM tries to move the yellow dots sort of a little bit more together, than they're less dispersed now. And then lasso ESM further like breaks them. But maybe the 2 dimension is not enough to represent the lasso versus non-lasso. Just 2 dimensions is not enough. If we go into 3 dimensions, also we can separate the orange and the blue points. Okay. We could do a lot of validation for these things. We generated like 2 different sets of libraries were present, where we could look at 3.2 million samples, where we are looking at these the sites that I've shown you here, 7, 8, 10, 11, and then this 2, 3, 4, 13, 14. What we were able to do is you can generate a big pool of all possible variants of these for these sites, and then you can predict whether and you can test some of them further, whether they are a substrate or not a substrate. And you can do all of this through cell-free biosynthesis. So this is all work done by Doug Mitchell's group. So they can get very reliable predictions. So as I said, that we use active learning in this, where we make the model, we predict new samples, they test it, and we also try to look at whether we can beat the experts in the model. And so we select the mutants that have very high uncertainty, and so that we can get more informative predictions. And so every round we were testing like 60, and some mutants were tested. And then you can see that with just like few rounds of very informed experimentation, we were able to actually go to a very high accuracy, model for predicting the enzyme specificity of. We have done, I mean, I've not talked about that. We have tested this model on a lot of different things. We can predict lasso, cyclase, and peptide pairs using this tool. We have actually tried to predict the antimicrobial activity of the lasso peptides as well. So we can get variants that have high antimicrobial activity. And all those test cases are actually in the preprint. We've also done this on another class of enzymes, for which we have an mRNA display data. So this work was done, was sort of a collaborations with the Hiro Suga's group in University of Tokyo, where they had actually already published an mRNA display data, and they were able to share that with us. So what they did is they did an mRNA display so they can find out the substrate for this particular class of enzymes called LASBF and LASDEF. And they used a CNN and as a deep learning architecture. And with millions of sequences that they were able to test. They were able to train a very high accuracy model. So where we come in here, so this paper was in ACS Central Science just like a couple of years ago. We were able to show that, if you use a peptide-specific language model, and you try to now do the predictions, instead of millions of samples, you actually need just like tens of thousands of samples. So roughly around with 10,000 samples, we are able to get the same accuracy as when you use 9 millions and you use a CNN. So the language models are actually allowing for transfer learning between different sequences. Uh, and that actually gives you the same accuracy with very limited amount of data. So this was also very nice. It was a very short paper, but it had a very, so to say, very strong conclusion that just like 1% of the data would have been enough to build a model with exact same accuracy as the standard, model, including millions of data points. Okay. So I'll just give you, few things here. Um, we were able to train a lasso peptide specific models. You can use, if you can make a pre-trained language model that helps with the transfer learning, that helps with the limited data. If you can use active learning, that also can reduce the experimentally labeled samples um, you know, that you need, right? That's the key message. And now we are using these models for designing lasso peptides, targeting, different types of GPCRs, and that's like our current work. Um, Finally, I think I'll give you. I have maybe 10 minutes, and then I can take up all the questions. So I'll just give you one quick example of when you have no data, essentially, or one-shot learning, as it is called. Uh, can we predict the protein ligand when we have very, very limited amount of data, and how can we leverage all the different, uh, uh, how can we leverage different proteins from the same subfamily to actually train a one-shot predictor? So, here we were mainly focused on GPCRs and kinases. These are the very important drug kinases. But if you look at it, kinase data, 51% of the kinases have less than 50 known inhibitors. If you look at, and 16% have less than 10. So still, there's a need to actually predict substrate for the kinases. GPCR data is very, very similar. So there are a lot of known ligands for, uh, even if you look at, and there are very, very few receptors with greater than 50. So most of the receptors have very limited number of known ligands. So we took all these data sets, we cleaned them up, we identified unique GPCR ligand pairs and try to train a model. So this is called one-shot learning, and this is like a very popular. I'll just stop. I'll quickly tell you this thing. So the idea here is that that based on the different GPCR or kinase, whatever enzyme you're looking at or receptor you're looking at, you can look at these substrate enzymes pairs, and from that information, what you're doing is you're comparing your query molecule to all the available pairs, and then you are trying to get an average similarity between the query and the different types of entries in your class. And then there is also an attention mechanism built in that actually serves as a weight on, you try to weigh in the molecules that are very similar, or the substrate or the enzyme proteins that are very, very similar automatically. So here there's an example that you have pictures of 4 dogs, and you're trying to compare a query picture of a dog and can classify which type of a dog it is, right? So, that's a key idea, that with the single picture, you can actually classify something easily. So we use the different types of one-shot learning architectures. The one is known as the Siamese network, and then the other one is the attention LSTM. I'll not go into the great details of this, but these are popular one-shot learning frameworks. And then we can take different GPCR ligand pairs or a kinase ligand pairs, and then based on our query, we can actually try to predict whether with just a single information or no information, we can actually predict whether a ligand will bind to a particular GPCR or not, or a particular GPCR will bind to this ligand or not. Like both ways you can do your predictions. Okay, so this is the model and this is the accuracy. You can see that these are all for the kinases. There's a very uh, we are getting a very, very high accuracy model. I think the data set was a little bigger, and both the models were doing a really reasonable job. For the GPCRs, you have a lot more diversity in the binding pocket. So basically, your support set here is very diverse. So you're not finding a lot of entries that are very similar. Kinases have a pocket that is very conserved. So you can leverage the shape of that pocket information there to look at ligands. But for GPCRs, you have so much diversity. You're binding peptides, you're binding, lipid-like molecules, you're binding all sorts of hormones, neurotransmitters. So there's a lot of diversity in ligands in GPCRs. So I think that led to a little bit lower, accuracy for that. So again, this is just to give you the same idea that GPCRs are binding so many diverse ligands, so it gets a little bit tricky. But if you look at the kinases, there's a very high conservation in the pocket. So it was we can get a very high accuracy prediction for that. So we actually wanted to test this on completely unknown compounds. Now this is becoming a really popular topic of discussion. We tried to look at whether the drug excipients, these are the molecules that are supposed to be non-active molecules that are put in drugs. And we wanted to see whether the drug excipients have any activity towards these signaling molecules. So, this is one example which I'm showing is the EGFR kinase, which is involved in cancers. If you look at it, what we are finding, this is the known drug that binds to the GPCRs. But now you're finding that all these dyes that are used in food products all the time, they can actually, very reliably bind, they're predicted to bind, and then we do docking. We can also do, for some of them, we also did the free energy perturbations. We actually computationally predict the binding affinity. And you can find that all these dyes are actually binding to these key signaling, receptors and in our body. Okay, so that's one thing. And then so one-shot learning is essentially it's a very good model where you are using all the pairs of substrates and ligands that are available or known in literature, and then you can leverage that information for completely unknown ligands and receptors. And for the two classes that we showed, that it's a fairly reasonable models. Now, I think the last leg, I'll take 2 minutes and I think I'll be done. I'll give you one last example of the current work where, in the beginning, I talked about cross attention, where I said, okay, you can learn the interaction between the enzymes and the substrate pair and substrates. And I think that's very, very useful. Um, but then what happens is you have to actually train a model to do it, right? You have to have a cross attention built in, and then you train a model. Why not just do it beforehand and give it to the people? So, for example, if I go to my next slides, you have protein language models and you have chemical language models. The language models, I think everybody now knows what these things are, but this slide just shows you that the structure, the sequence, the tertiary structure, quaternary structure, these are could be represented as a language of proteins. Similarly, different chemical groups, if you represent them as smiles, you can think of it as a language. But then the point is, what is the interaction between the proteins and the ligands? Or proteins and peptides, can that be leveraged as a language? Can a language be not just protein or not just ligand? Can a language be an interaction-based language? So I'll give you, we have a couple of work that we are just finished on this. I'll give you one example from our work where instead of building the cross attention later on based on your data, you actually put it at the front with the language models, and then someone can have those embeddings which already learned about the interaction. And then you can very quickly build a very small model on your laptop train it on your data and actually get limited data, and then you can actually get a very good prediction. So I'll give you one example of how we combine them. So there's this idea called composition, is becoming popular now. This was 2024 paper from Google DeepMind. Um, and the idea there was, just to give you a very simple example, is that we have one language model that is actually learning, key value mapping. The idea here is that x1 is equal to 10 and x2 is equal to this and x3 is equal to this. There is a language that connects symbols to numbers, for example, right? So the relationship between symbols and numbers can be learned by a language model. Similarly, arithmetic could also be learned by a language model. So, there's another model that can learn arithmetic. So it can add two numbers and give you the correct results, and very, very high accuracy with very limited data, you can train these models. But then what happens is when you try to do symbolic arithmetic, where you want to add x1 plus x2, then both the models actually don't do that well, right? Because the symbolic, the one who knows the symbolic key value mapping doesn't know anything about arithmetic, and the model that can do arithmetic doesn't know how to deal with symbols. So that's a quick example where now you can take the layers of these models and you can combine them using an attention mechanism, so that it can actually do symbolic mathematics. And this was a very interesting idea. And you can see that that the combined model can do both symbolic assignments and also can do arithmetic. It's a fairly accurate model. So we can do the same thing with peptide protein language model, peptide ligand language models. So the idea here was that, for example, you have a peptide protein complexes, and can we predict the residues? We train this model based on which amino acid is interacting with which amino acid in a peptide, and can you predict that? So you have these contact matrices between a peptide and a protein. Can we predict these contact matrices? So we have this attention mechanism between a protein language model and a peptide language model that actually learns the contact maps between the proteins and peptides, and then it gives you an embedding. And surprisingly, what happened with this model is that you actually don't need, you know, thousands of dimensions to represent the interactions. We are actually able to show that instead of 1280 models, 1280 dimensional model for protein language and another 1280 dimensional model for the peptide, you can actually just make a 128 dimension model, and you can actually learn the interactions really well. And it outperforms, the individual models um, uh, substantially. So here is an example on like 6 different classes of proteins and their substrates. And you can see that the red bar, we call it PEF fusion, is the name of the model. The red bar actually is pretty good for most of the cases. It also depends upon the amount of data that is available. Sometimes when there's very limited amount of data to train, um, you can simply concatenate the embedding, and that also works in some cases. But in most cases, you can see that we have a very, very accurate model for predicting peptide protein binding specifically. Okay, with that, I think I'll just end. Uh, what could you do? Automated data generation is good. Augmenting experimental data with computational data is good. Active learning could help you. One-shot learning, I gave you an example. Transfer learning from protein language models. And then if you can leverage the interaction information, which is missing from standard language models, I think that actually helps a lot with the I think with that, I think I'll just end. Most of the work here I showed was done by 2 people, Tanner Dean, who is a 4th-year graduate student, and Xuan and me, who just graduated and works at Johnson & Johnson now. Um, and a lot of experimental work were done by Kim Zhao and Doug Mitchell's group. And this is all funded through NSF and NIH. Okay, I can take the questions. I think.
Thank you. Thank you so much. That was awesome. If you could go through the questions in chat and read the question out loud before you answer it, that's going to help with the people that watch the recording because they don't see the chat.
So the question is from Jose Perez, which is, can you use the ML pipeline to predict reaction catalyzed by non-canonical catalysts like peptides with unnatural amino acids? So that's also a very active area of research, that we are working on right now. If you want to use unnatural amino acids, The the big challenge here is that we don't have any information about unnatural amino acids in the language models. Why we don't have any information? Because the language models are all trained on the sequence data that does not include unnatural amino acids. So what is the way to get around this problem? And I think that's what we are doing right now. And there is some interesting work from, I think, from Duke University. There is there's a professor named Pranam Chatterjee. He does a lot of work on peptides, and he came up with, and he has done something similar, and we're also working on similar idea in different contexts in my group. The idea is that if you can somehow represent the unnatural amino acids as chemicals, and the rest of the peptide sequence is a peptide. So you represent your peptide both as a chemical and as a peptide sequence. And if you can combine somehow these informations from chemical language and the peptide languages, then you can represent unnatural amino acids. So then you can get embeddings that are that can represent unnatural amino acids, and then you can train your models on that type of an embedding, and hopefully that can deal with this type of a problem. I think this is just an idea. Not much work has been done, but I think unnatural amino acids is like the next thing on the list for so many people. Okay. How do you use, there's another question from Aditi Biswas, which is, how do you use VinaDoc to predict doc with ligand like ferredoxin and FES cofactors? So we have actually tried to predict the cofactor is already bound to the, we tried to predict the structures with the cofactors bound. We were able to do simulations to relax these cofactor bound structures. And then we tried to put in a ligand into the pocket. So there was a lot of refining that was needed to be done for enzymes that have a bound cofactor. You can also imagine in terms of in halogenases also, you have different types of halogenases with different cofactors bound. So we had to do a lot of modeling as well, molecular modeling here to get a reliable structure for the complexes. So I think that's how we dealt with it. Um, Megan Jackson has a question. Do you have a way to account for eternal conditions, external condition? I think that's what you're going to say. External conditions like pH or solvent, or those are parameter baked into model that you can't easily change. I think we are like tied down by the data. Um, uh, it's a little tricky to account for all those conditions. Um, there are there is a lot of interesting work that is going on right now on predicting the optimal pH and solvent, I should say. We have within this center that we have called Molecular Maker Lab. Um, uh, there are a lot of models that are getting developed, and I can also see people have predicted optimal pH for certain reactions. But I would say the data that we have from literature, they are all, what should I say, um, they are all at a particular pH. We have not explicitly accounted for it at this moment, but I think it will be very, very important for when you're predicting the reaction rates like Km, Kcat. I think all these external conditions become very, 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 very important. So, people who are developing these models, and I know a lot of people here who are trying to, students who are doing it. So they are taking these factors into account, and you need some very clever techniques to augment the data um, to build those models. I think I'll just say that. Um, in the data mining process, did you prioritize smaller, high quality data sets, or was your approach more focused on using large quantities of data? What is the best approach when using AI for predictions? Um, I think this question has been, I think the correct answer is it depends on the way the data is generated. Um, Even if you make high quality measurements, but if the data has a lot of redundancy, then you will not actually learn anything. I'll just give you one simple example. For example, when we are looking at proteins and you're doing like a mutational scans on protein, like that's a very popular technique now. So uh, if you, let's say, it's better to have some information for all sites of a protein than having all the information for some sites. That's a heuristic that you can sort of think of. Like if you have a broad data set where there's a lot of independent data points, that is much better than having a lot of data points, but they are all repeating themselves. I think this is a very common thing. You also see this with sequences. Like if you build a like a multi-sequence alignment, but if all the sequences are exactly the same, then there is no information. So I think it's not about the size or the um, of the data set. It's more about how much independent information is available in the data set. Okay, that is much more important than the number of data points. You can develop a heuristic for the number of data points as well, but my thought is the information content is much sort of an informative way of thinking about the size of the data sets. Octavio, what kind of self-reference system did you use to make the protein tested in your model development? I think I will defer this question. I cannot really answer this question accurately. I know what has been written, but I will just say that um, this probably is for my collaborators to answer the self-free system side. So I'll skip this one. Oh, Tai Sok Moon has a question, which is the difference between protein ligand binding and enzymatic reaction is that the enzymatic reaction prediction should be approached from the transition state structure that is difficult to predict. Thus, binding prediction would be insufficient to design good enzyme. I completely agree with both the points you just said. Okay, and then the following statement, which is similarly, transcription factor engineering face similar changes with difficulty of predicting conformational changes after ligand binding. Could you comment on those points? So number one, I completely agree with all the observations in this question. Um, and that is why, I would say we have an MMLI 2.0 that is getting funded by NSF, hopefully, if things go well and with funding. The transition state prediction is what we are trying to do. There is very limited amount of, there is already a transition state predictors for solution reactions in solution between chemicals, right? Few groups have done it, but there is nothing available for enzymes. So what we are doing, what is used then as a proxy for the transition state essentially is what is your distance from the different, when you have a ligand bound into a pose, what is this distance from the different reactive residues, for example. And you can build a very simple model which can give a reasonable prediction of reactivity by just having an accurate, I should say, product bound and the reactant bound conformation. If you can generate that, I think you can predict it. At least for one class, we were able to do this, which is a particular class of halogenases, where we assured that uh, the reactivity was directly proportional to the distance from this between these two atoms in the model. Uh, but it's a very hard thing to get those things accurate. So we have a transition state prediction work that is also sort of going on. It's a very challenging one because you need a lot of DFT data for it. So, and that's slow and to generate probably take months to a year to even finish that type of a data set. Um, so that work is going on and it's on everybody's mind. Uh, Conformational change is much more interesting. Conformational change is much more interesting. Um, Predicting the conformational change, there is a lot of work that has happened on predicting the conformational changes of the protein, but it is basically everything is right now is based on the subsampling of the sequence. So you subsample the sequence. Basically, in a way, what you have done is you've introduced entropy into your sequence, and then you're using these sequences to generate new structures. So you perturb your sequence, and then you predict a new structure, and then that way you can generate different conformational ensemble. But there is no work that is being done on a ligand-bound conformational change. That is that if you have a sequence plus a ligand, then the conformational change is different from when the ligand is not bound. I think that part is sorely missing. Uh, what we have been able to do, and this work will come out, hopefully will come out soon. We have a model called ESM Dynamics now, which actually predicts which predicts the key residues that move on ligand binding or on the perturbation of the any type of perturbation that is introduced. So what it can do is it can take a sequence of the protein and a model. Uh, and you can also include in that model a ligand, for example, that is bound into the pocket. Uh, and it can try to predict which residues are going to move when under this condition. Uh, it's a I would say it's a fairly accurate model, not very high accuracy. We are not trying to predict the structure. We are just trying to predict the residues that move, and it gets at least the residues that move right. But I think the next goal is to predict the structures with these type of constraints. And just to final comment would be, I think we need to add some type of a physics into the ML models to actually capture these effects. And the question is, how do you add it? You add physics from a lot of simulation data, or you try to add physics into the architecture itself in some way. That's an open question. But these are all very interesting questions that I think we don't have. These are all research questions for future. Okay,
Yeah,
with that, I will stop.
thank you. The reason I asked those questions was the more than 20 years ago, I was so excited when I see the novel design of protein that has enzyme activity, but still very low activity. But 20 years later, still we are sort of struggling in terms of
Activity.
designing the new enzyme. That's why I'm asking that question. Second question I ask because I'm very interested in biosensor development, but biosensor typically rely on transcription factor that after binding the ligand, they change the conformation that now have ability to bind to the DNA differently. But still, those prediction is very difficult. There are only a few exceptions. Some people, including Tanya Kotemi lab, UCSF, published one great
One
paper,
paper.
but a few years ago only. So it's been more than 2 decades, but still we are. I mean, of course, AlphaFold, those kind of, algorithm kind of advanced significantly, Rosetta, helping us a lot, but still, more thing to
More work.
achieve.
I completely agree. I think I'll just give you one paper that one work that I didn't have a chance to talk about, which is also transcription factor, which is this is some work with that will come out soon with Mike Jewett's lab at Stanford, doing biosensor development. And there we were actually able to use a new algorithm. We don't even have a name for it. We call it is based on our group member's name right now. So, that algorithm, we are able to actually design a lead sensor from a sensor because, in water, there's a lot of zinc, maybe 500 times more zinc than lead. So whenever you try to sense like a +2 charge, any ion with +2 charge, it binds. So how can you make it specific? So again, with, I would say, limited amount of experimental data, we're able to completely shift the specificity to lead. Now you have a very high, highly accurate lead sensor that is, at the levels where EPA wants it to detect with zero interference from other +2 charges. So although, there is a downstream component to that as well, right? But if we don't bother about the downstream components and just focus on the function, just read the function and then try to change the sequence, somehow the black box is able to learn the effects.
I
But
see.
predicting the conformational change itself is, I think, more challenging.
Yeah. So I also pretty much benefit from that prediction model. So we used to use directed evolution for many years, but now we use the AlphaFold, Rosetta, or whatever prediction model. In that way, our process become much faster. So now I doing some protein engineering, but when I graduate student, I hate protein engineering because I need to pick the colony more than 20,000 by becoming robot and then pipetting the same amount. That's not fun. So didn't want to
Yeah,
do
yeah,
protein engineering.
I will see if I can share. This paper will be on bioRxiv maybe in another like 7-8 days.
Good,
So I will share it with you as soon as we put it.
sure.
That might give you some, might be an opportunity to even work together.
Yeah, we also able to change the, substrate specificity. In that way, we develop, great sensor, we publish a couple of paper about that one, but still, I'm learning in terms of computation.
That's,
Thank you. Fantastic.
thank you, Kate, for the invitation.
Yeah. Thank you. This was an amazing talk. It really inspired a lot of discussions and a lot of ideas possibly. Thanks again, and thanks everyone for a great discussion, and have a great rest of your Monday.
Thank you.
Bye.